So, you're at HBS and you have this wonderful Nucleate experience. You're connecting with, you're seeing it kind of like in action in a place where you're like, okay, maybe we should do this for real. Talk a little bit about that point, that journey of like, we're gonna do this for real. And like the
Yeah.
co-founder, like how did you come together with your co-founder,
Yeah.
the company origin?
Yeah.
Talk to me.
Yeah. The origin story finally. And not finally, it's really important because part of KiraGen is my relationship with Ryan and all our experiences. So KiraGen formed from a pitch deck exercise. Kid you
Yeah.
not. So we talked earlier about the learnings. We had a lot of learnings and we became best friends at Bluebird and Beam because we were always asking each other, why are we doing this? What is the strategy here? And now looking back on it, we were forming what we would do if we were ever given the chance to do things on our own or differently. And I called Ryan up. So we worked at not only Bluebird together, I was an early employee at Beam Therapeutics, brought Ryan over once we sort of evaluated base editing and T cells. So our backgrounds are cell therapy at Bluebird, getting to see transformative outcomes for CAR T in leukemias and some lymphomas, but challenges translating that to solid tumors for many different reasons. We were already kind of getting our feet wet there, but really see again the benefit of frothy markets. We saw so much science, and it was really great for us to get access to that, but also learn that we need to also be able to know how to move things forward and make decisions. And not that they didn't, but that was something we really wanted to clue in on. And how would we rationalize decision? At Beam, it was a transition of now you could go into what we call CRISPR 2.0 and say, now that you're not a genetic scissor, you're not a nuclease cutting, you could use base editors or now others to what we call the pencil and eraser at Beams. But and it's great from now. The company's really focused on in vivo disease correction. It's an amazing application for it. For cell therapies, it actually allows you to think about how to flip the script on how to design these modifications on cell therapies that maybe can imbue it with different functionalities that an artificial receptor or something else with a nuclease cannot do. So at Beam, this was multiplex editing. So you can now make many modifications without really altering the viability or health or having these genomic sort of aberrations by making too many cuts. At Beam, we learned, okay, you can make many modifications. How would you apply that? And we did it with a multiplex edited CAR T for T cell leukemias. So this is important because you have to not only have a CAR, you have to remove the surface molecule that the CAR is targeting because it's targeting T cell leukemias. It's a great application that opened our eyes to say that was just the beginning. What happens when we think about moving beyond? This is a quad edited product. That was the first time a healthy donor derived, so it was allogeneic, edited product. It now is in the clinic. It's led to some CRs, amazing time. What happens when you start installing multiple edits? When you take like a George Church, David Liu paper saying we can make 200 edits in a cell, what do you do with that? And that was part of Kieran's story. And this is nothing against that. The tech now, so talking about tools, the tools to create these really cool things is not the bottleneck. The bottleneck is application and the people, and how do you make something that people should care about and can be developed to take into the clinic? So when I called Ryan up, he had gone to get his PhD, really focusing in on can you go in maybe a different direction, use multiplex editing to think about solid tumor microenvironments, which are low in oxygen, have all these things that are upregulated to turn your immune system off and our immune cell-based therapies. Called him up and said, hey, like, let's put pen to paper on something that we think is cool using our expertise. Is it still differentiated? It's part of the pitch deck exercise. It's like MBA 101. Is your idea differentiated? Do a landscape. Then the best thing is to build in something new to the company. So long story short, it is new. 'Cause most people that are working with nucleases or these sort of artificial strategies, if they are encountering a suppressive microenvironment, they try to make these T cells or NK cells more potent. So you're trying to break through the suppressive barrier, which is fine and dandy and will work in some applications. It probably can't and should not be applied in some sensitive areas like the brain, because the inflammation in the brain is really not tolerable. And we said, okay, that's one thing, but you're still redirecting function and compensating for these sort of signals that a tumor or all these other suppressive cells are spitting out at your cell therapy. So you redirect with a cytokine or you try to change the potency in a different direction. People are doing this because they were limited maybe by the 1.0 generation. We are not limited by changes. So let's first principles approach. What's the problem to solve? Let's not do what other people are doing. What's the problem? T cells, NK cells, gamma delta cells, whatever it might be, sense these negative signals from receptors on the cell surface. And there is a way you can knock out those receptors only and actually create this, what we call TME suppressive protection layer that just prevents the cell from being suppressed. That's really the crux. Beyond some physical barriers, which are still present, there are a lot of examples where a T cell can get to the tumor site. It has the ability to also address tumor heterogeneity via next-gen targeting strategies, and it gets turned off almost instantly. It might start signaling and turned off. There are 50 to 80 different pathways in most solid tumors that are upregulated. So if you are a PhD student and you work on TGF beta, which is a common suppressive factor, and your tech is one receptor that's going to block TGF beta, that is really cool, but it's not going to be enough. Our thesis on this pitch deck was that you need to address multifaceted TME suppression, just like approaches in the antiviral space, in antibiotic space. You need to really shotgun approach it almost to address many different non-redundant pathways that usually tumors use to evade whatever strategy you're after. You can't just block PD-1. The biologic space is now challenged by not being able to give people anti PD-1, CTLA-4, LAG-3, because you have toxicities from these molecules. Well, for us, we prevent suppression before it happens. So there are ways that are not trivial, which is part of the second part of the company, to knock out just the receptors that don't alter potency, but allow a function that we know works in heme, at least to some extent,
Hmm.
to finally translate to solid tumors because the CAR-NK, whatever it might be, can get there and continue signaling consistently, persistently. Durable functionality is our goal. So not only is the biology of making this many edits very counter to where the field has gone in the past, people said, how would you ever decide? If I told you that our lead product is a 6 edited CAR T cell, well, for us, it's not screening. We are design optimization, and now we have a machine learning model that rationalizes that for us. But it's not screening from scratch. This is a huge shift of every company needing their own novel protein, a novel pathway to block. Our protections around the combination itself and how we're protecting these cells against suppression. And we've been able to rationalize it from this list of 50 to 80 down to, say, 6. Very clinically validated via things that we know in the clinic to work. It's biologically sound. All the regulators know the pathways, but no one's had an approach to address suppression, but to a way to make you care and believe that combo is what should be going into that space. So that came together. I'm getting back to the entrepreneurship and we'll talk a little bit more, but we were just an idea. So we made a pitch deck. And then when the exercising class ended, we kept working on it. We were like, this is cool. And 2048 Ventures, who has a biotech studio program that is very similar to Y Combinator, but very focused on biotech, gave us our first capital. And they believed in our idea. It was well vetted, of course, from our background, but gave us a set check size. We filed IP. We got into the lab on campus. So the Harvard's Pagluca Life Lab. As a student, you get free access to like a shared bench, which is super helpful. really great. And now that we're a real company there, you get some subsidized help, but also this infrastructure where you don't have to buy everything from scratch. But when you do need specialized equipment, you can go to Exceeder and and lease things that we know are our workforce assets. So it allowed us to get in and be capital efficient, right? So we were doing something that still to this day is very counter to what people think. People spent money in cell therapies and they are running to either in vivo or ADCs that have other advantages or selling points about being cheaper or whatnot. We said we can do it differently. We have this experience. What we do is using validated processes, reagents, and methodologies, but we're putting the puzzle pieces together in a way that has not been done before. But we are not the only, we are building upon science in the past. This is important because we do not focus on binder design. We do not focus on all these other things that maybe we have to partner with someone on, but the solution to solid tumors is not going to be one thing. Just like our CAR T cells are not one piece of technology, we need to be building upon others. Even if we had all the money in the world, our lead program is in glioblastoma, which is a extremely aggressive and rare brain cancer that has very little progress has been made to date in the last 20 years getting therapies that really help patients. It's inevitably a death sentence, but in the brain is where all this progress in CAR T has been made to date, which people don't really think about. A lot of cell therapy success has been very little in the solid tumor space. In glio and some gliomas, there has been activity, like actual activity that's measurable. We are working upon others to build finally upon clinical, not only clinical success, even if transient, but we are addressing known problems. So not only do we have this kind of, we have a crazy contrarian sort of biological approach, which is this multiplex editing, but it's grounded by solving known clinical problems that have already been identified to durability in these patients. So it's not 0 to 1 anymore, which people don't really see. It actually is, it's 1 to 2. And what we've found, which is exciting, we found a really perfect product market fit as an early stage company, but it was timing. It was a pivot. We did not start with this. Nucleate helped along the way, and I have a story here about that. But the perfect product market fit, but in an indication that people are scared by, rightfully so, if you're a VC or other funders, nothing has worked in the past. And so it is a product market fit with an indication people might be scared of, and also in modalities like ex vivo cell therapies and gene editing that aren't super in vogue. And this is a great lesson for anyone out there, but it is our background and expertise. We did this company because we didn't see anyone else doing what we were doing, and we didn't think people would do it. And now, if you can weather the storm, if I can get 2 minutes with you or on this podcast, if people are listening, to tell you why, if you can, for a minute, take away the historical challenges of glioblastoma, it is perfect product market fit. We're addressing challenges that are known problems to durability, not functionality, not efficacy from getting 0 to 1. And so that gets us really excited. We can not only, we found a spot to not only thrive, other people are not in, but also this non-potency enhancement based approach is best suited for the brain and other spaces for now. It's actually a great sort of proving ground where there's not a ton of competition yet. And we think it's actually biologically differentiated so that when we get to that first in human, we can spread in tons of different directions because most tumors have these suppressive environments. So lesson, I'll wrap that up, is know as a founding team what your skill set is. Even if everyone is going into in vivo autoimmune CAR T, which is amazing, and every tool should have its application, doesn't mean you shouldn't be doing what you're doing. People will tell you to go in other directions, and it's usually best to stick to your guns and do what you think is best for you and the company.